Welcome back, guys. And in previous videos we have talked about the 5 prime capping, which is a part of the post transcriptional, or not post, actually co-transcriptional modifications. And in this video we'll be talking about another type of very common co-transcriptional modification, and it is called 3 prime polyadenylation. Okay, so we'll be talking about 3 prime polyadenylation. So as the name suggests, after the mRNA which is produced, it must have two directions. So one is 5 prime, another one is a 3 prime. Remember, so we know that this is a DNA, and this red colored thing is the RNA which is produced. Rather, you can call it as a mRNA which is produced, which is actually producing from this DNA. RNA polymerase 2 sit onto the place, it is start transcribing this mRNA is being produced. Now this is called the co-transcriptional modification. So as the term suggests, co-transcriptional modification, that means this type of modification happens along with the synthesis of mRNA. Okay, so as the mRNA is producing, this type of modifications are simultaneously happening. 5 prime capping has happened just after the production of 5 prime region, because this is the direction of mRNA synthesis. It's always from 5 prime towards the 3 prime. So it is being synthesizing from the 5 prime direction. So after producing 5 prime end, 5 prime capping is done. We have seen what is capping, what is its importance, and the mechanism of capping in our different videos. Now in this video we'll be looking at the 3 prime polyadenylation. Now one question can come into your mind. We are calling it co-transcriptional modification. So this 3 prime capping must also have done during the transcription process. But during the transcription process, we must have the 3 prime end of this mRNA to further modify it, right? We can only get this 3 prime end when the mRNA synthesis is completed, isn't it? Whenever we are getting it completed, then only we can modify it. This is the question. The answer is yes. Until and unless we get the whole mRNA like that, so this is 5, this is 3 prime, then only we are able to modify this 3 prime. Otherwise, in this case, it is producing, so we cannot find the end of 3 prime to be modified. But it is also called co-transcriptional modification because the modification procedure begins during the processing of mRNA. It begins, but the actual termination of the process will only be achieved whenever the mRNA is completely produced. Okay. Now in this case, we require many different varieties of proteins. There are different types of proteins, but the functionality, we need endonucleases, we need this RNA binding proteins, we need some of the specificity factors that are associated with this mRNA, which can help to modify this mRNA. Okay, and also we need polymerase, which can add this adenine, because it is a polyadenylation. That means basically we are actually adding adenine at the 3 prime end of the growing mRNA. So this is the modification we are looking about. So that is why it's called polyadenylation. So many adenine residues are added at the 3 prime end. So we must have access to polymerase. So from here we can say we must need polymerase. Okay. Along with them, we need some specificity factors. Factors like CST, factors like CF, C for cleavage, cleavage stimulating factor, cleavage factor, cleavage and polyadenylation specificity factor. Like these are the proteins CST, CF, CPSF. These proteins are required for the accomplishment of this process. Okay. So why we need polyadenylation? First let's talk about that, then we'll go to the mechanism. We need this polyadenylation again to protect this mRNA to being cleaved by the exonuclease from the 3' end. We have already protected, remember, this polyadenylation events always occurs after 5' capping, because 5' is free from the first time, right? Because 5' is produced in the very beginning, so 5' region is free from the beginning, so it is being modified. So we have seen, say this is the capping, so we have already have 5' capping, and we protect 5' to be degraded by the enzymes. Now once after fully transcription of the mRNA, we must modify the 3' end also, otherwise this can be degraded in any aspects. If it is degrading from 5' or 3' end, the actual goal of our mRNA to hold the codes to produce proteins will be disrupted. So that's why we need to protect this mRNA from both the directions. In the 5' end, we protect it via 5' capping utilizing guanosine residue attachment. We are protecting it being cleaved from the 3' end by attaching polyadenylation, by attaching polyadenine residues. Okay. Now here this process. Now how this polyadenylation can give the mRNA stability? This is a question. Say if exonuclease works on it. Let's say exonuclease works on it. Now it starts cleaving one nucleotide at a time. We know that this is a feature of exonuclease, that it cleaves one nucleotide at a time from the terminal ends. So cleaves this A out, then cleaves this. So this polyadenylation stretch, remember, it is not required for the protein synthesis, or this 5' capping, it's not required for the protein synthesis. So whatever we are adding here at the 3', it's just for protecting it. So what we are doing, we are providing some junk things. Now if you want to cleave it. I don't bother if you cleave this adenine. That's the actual goal. Cell won't bother if it cleaves 1 adenine, 2 adenine, 3 adenine, 4 adenine. Let them cleave. Just cleaving, right? We won't bother, because from this point we are having the coding region, but the rest of the part is not required at all. And usually there are 200 adenine residues are added. 100 to 200 adenine residues are added. Normally it is seen. Okay, so 100 to 200 adenine residues, it take a long time for the exonuclease to cleave, and before that this reaction would be accomplished. This is the first point, how it protects it. Second point is that there is a protein called, I haven't write it here, the protein is called PBP, or polyadenylation binding protein. Okay, or PABP you can also write it. Now this polyadenylation binding protein, or PABP, can bind with this polyadenine residues. Now this PABP in turn can take this 3' polyadenylation region and 5' region, attach them to produce a circular DNA molecule, circular RNA molecule. Okay, so utilizing this PABP, what they can do is that, say this same structure, it is, so say this is polyadenylation is from this place, polyadenylation region, and this is the PABP. It is holding this 5' and this 3' to produce a circular RNA. So we can have a circular mRNA, and having this circular mRNA can give us the advantage, because remember, exonuclease cannot cleave from the middle of any phosphodiester bond. It can only cleave from the terminal ends, so it gives the protection to the mRNA. So this is how this 3' capping protects our mRNA. Okay, it is a projected models. Now let us talk about the mechanism of 3' polyadenylation and the importance of these proteins and how they act to actually produce this polyadenylation. So let us draw this picture in bigger way. So I'll be drawing it similar way. So we are having the DNA, and also we are having this mRNA which is being produced. Now, when this mRNA is being produced, when it reaches a particular signal, the signal is called a polyadenylation signal. Now, when they reach the signal, remember I have told you before that it can only attach this poly A tail when the mRNA is totally transcribed. But before that, during the transcription, it starts. That means during the transcription, it encounters a region. It encounters a sequence, and the sequence is U. Sorry, it encounters a signal like AAU, like that. It encounters this type of signal. This is called polyadenylation signal. Okay, whenever it encounters the signal, polymerase encounters the signal, they recruit some proteins. Proteins like CPSF, cleavage and polyadenylation specificity factor. When they require this, when they get this specificity, they just recruit cleavage and polyadenylation specificity factor. Then it keep on transcribing. After some region, there is another sequence. It is CA sequence. So this is again 5 prime towards this. This is the 3 prime of mRNA. Okay, red thing is the mRNA, blue thing is the DNA. Now I am drawing this picture along with DNA because to show you that this process is going on simultaneously. This process of polyadenylation begins during the transcription process. It concludes only after the mRNA is totally transcribed. That's the basic thing. Now here there is a CA. After some time there is a GU sequence there. After further downstream there is a GU sequence. We don't need to talk about that. But whenever it encounters this sequence AAA, it calls on this CPSF or cleavage and polyadenylation specificity factor. Now that protein comes in. Along with that protein, CST or cleavage stimulation CST, not T, I guess it's CSF, cleavage stimulation factor. It will be CSF. It's not CST, it is CSF, cleavage stimulation factor. Sorry, cleavage stimulation factor. Now cleavage stimulation factor and cleavage and polyadenylation specificity factor, say let us draw, this is the cleavage and specificity, cleavage and polyadenylation specificity factor, and let's consider this as this black thing as cleavage stimulation factor. Both proteins bind with two different sequences. This is, so let me write it, this is CPSF, this is CSF. I don't know whether you are able to see it, so let me write it with different color. I think it will be better to write it with this red. So this is CSF and this is CPSF. Okay, they recognize those sequences. This first process of polyadenylation is the sequence recognition. After they recognize a sequence, they recruit cleavage factor and also they recruit polyadenylation binding protein. Along with that, they also recruit polymerase enzyme. Polymerase enzyme is sitting on there, it is transcribing, right? So we don't need to bother about that. There is a polymerase enzyme, so I am just drawing the polymerase here like that. This blue color thing is the polymerase enzyme. It is sitting onto it and it is start transcribing. So polymerase is here, so this is pol2, it is transcribing. So as this CPSF and CSF recognizes both signals, CSF recognizes CA, CPSF recognizes this stretch of adenine and uracil sequences, they recruit this CF or cleavage factor. Now this cleavage factor is nothing but an endonuclease. It's an endonuclease. Now this cleavage factor binds to it and it cleaves just right after the CA sequence. It cleaves just right after the CA sequence. That is the time for us to continue the polyadenylation process. Remember, in case of prokaryotic system, this termination of transcription happens in two different ways. One is a row independent manner, another one is a row dependent manner. In row independent manner, there is a stretch of AT-AU linkages right after the GC linkages. It produces the stem loop structure, it falls out. So palindrome like structure form, they produce the secondary structure between mRNA, and it will fall off due to the strain that is being formed. Now in case of this eukaryotic system, if we require to do the polyadenylation, we need to cleave it, and this cleavage is achieved by this cleavage factor, which is an endonuclease. Now this cleavage factor will cut this mRNA, growing mRNA, just right after this CA sequence. It will cut it, and as soon as it cuts it, it recruits the PABP or polyadenylation binding protein. So then what we get this is mRNA, and say here we are having say CPSF, then we are having CSF, then at the back of all this we are having the large polymerase. This is a polymerase, CPSF, and this is CSF, and then this also is attached with this CF, which cleaves it. So CF comes in, so CF, cleavage factor cleaves it just after the CA sequence. So at the end we are having CA sequence, it is cleaved by CF, CF will be released, and in turn PABP will be recruited. So another protein PABP is recruited. Try to understand this point. This is the complex which is formed. CF cleaves it out just after the CA, then PABP will be recruited and this CF will be released. Now after that, this polymerase will start adding adenines in this direction. It will start adding adenines. So let us again draw the mRNA. Here it starts adding adenine via normal polymerization process. It starts adding adenine. So let me draw this polymerase here. So this is the polymerase, starts adding, and it will keep on adding until it reaches 100 to 200 sequences. It will vary from species to species, and these proteins will be cleaved out, but in turn this PABP protein, PABP or polyadenylation binding protein, will bind with this polyadenylated sequence. So at the end point, polymerase 2 will be synthesizing it, and PABP or polyadenylation binding protein will bind with this polyA sequences. Then rest of the protein factors will fall off, and then it will end up with producing this mRNA. So now as this PABP is attached, now this PABP, so if this is a mRNA, this is the 5 prime, this is the 3 prime, this is the 5 prime, this is the 3 prime. Now 3 prime will be coated with PABP, and it will hold 5 prime, circularize this mRNA to produce something like that. Okay, so this is the machinery of how they are making it. So first, sequence recognition via CPSF and CSF, then the recruitment of CF. CF will cut exactly after CA sequence at the 3 prime direction, then it will generate the mRNA, then they recruit polyA binding protein or PBP or PABP, whatever you can say. Now then polymerase will start adding polyadenylation residues. Now for this we must have access to adenines, so we need a lot of adenine, and all this adenine we require will be at the form of ATP, and it will produce inorganic phosphate. So this polymerization that will be carried out at the 3' end is simple polymerization. So just the polymerization between 3' hydroxyl and the 5' phosphate group polyadenylation, pyrophosphates will be released, adenine will start keep on adding. This is a regular polymerization, so we don't need to bother about that. Then they produce this, and then poly A binding protein will bound with this polyadenine sequences. Then it can circularize it to further protect it from the degradation. So the basic point is that they utilize this process to protect this mRNA from the 3' end degradation. And but you see, there are also exceptions. There are exceptions like histone proteins. They even won't have this polyadenylation, but still they are very, very stable proteins. So there are questions that nowadays scientists are arising that this polyadenylation is not at all providing any structural role to this mRNA. Whether it is really giving the advantage from being degraded or not, it's a region of research. So some believe that, some don't believe that, but we are learning it, so we'll take it. The logic I've told you that before giving this polyadenylation, if they want to cleave it from the terminal side, we don't bother because we have lot of A's, and those A's will not required for us. But still a question can arise that why will a cell will lose that amount of energy, because it will take energy to add all these polyadenyl groups at the end, right? So this region is again a part of active research. And also another important thing I must tell you is that in case of some prokaryotes, yes, in case of some prokaryotes and also some organelle mRNAs, polyadenylation signals degradation. So poly A, it can suggest degradation, it can suggest stability. This is the question mark for the new scientists. So you guys going to work, we going to work to find out whether which one is correct, whether this polyadenylation gives us the stability or it is giving us a degradation property. Because in case of some prokaryotes and organelles, this polyadenylation signals triggers the degradation of the mRNA. Okay, so giving this controversial thought, I am ending this topic, and I hope it will help you to understand polyadenylation. Thank you.